Hi, good afternoon. Today we have a very distinguished visitor that gladly accepted the invitation. Thank you for that, Scott.
My pleasure.
Yes. And is Dr. Scott Thompson. He is a professor of psychiatry at University of Colorado in the School of Medicine. And he received the bachelor degree in biology with a concentration in neurobiology from Cornell University and a PhD in neurobiology from Stanford in 1986. He spent then 10 years on the faculty of University of Zurich in Switzerland at the Brain Research Institute before coming to the University of Maryland. He was there faculty for 25 years and served as a chair of the Department of Psychology from 2011 to 2022. Then, last year, he moved to the Department of Psychiatry in the University of Colorado to establish the Center for Novel Therapeutics to advance translational research from the laboratory to the clinic for the treatment of neuropsychiatric disorders. Scott's research is focused on understanding what goes wrong with the brain in patients suffering from depression, and using this knowledge to identify novel therapeutics strategies for treatment. He focused on how stress is affecting or is a risk factor for depressive disorders, and his laboratory uses chronic stress to produce changes in behavior of rats and mice that are analogous to the behavioral symptoms of human depressions, such as anhedonia. So he has the findings that lead to the identification of novel class of compounds, negative allosteric modulation of GABA receptors that produce rapid relief of symptoms of depression in rodents, to novel approaches for increasing the utility and lowering the cost of using psychedelic compounds in psychiatry. He has several patents issued or pending that have licenses for 2 companies dedicated to bringing these compounds from the laboratory to the clinic. So today we have the opportunity to hear about his basic and also his translational research. So thank you very much for this visit, and hope you enjoy it. Thank you.
Yep, I think we're working. So thank you very much, Dr. Morales, for inviting me, and it's a great honor to be here with you to celebrate 30 years of this esteemed institute. And I'm delighted to share with you today some of our work related to psychedelic compounds. And what I'd like to do is really sort of three chapters here. One is to just start with a sort of overview of where we are as a field right now in using psychedelic compounds to treat psychiatric disorders, how the trials are constructed, what some of the clinical issues are, and then by way of understanding how these psychedelics may exert their beneficial actions, I want to take a step back and give an overview of what we understand as the neurobiological basis of a variety of these psychiatric conditions, particularly depression, and how antidepressant drugs treat depression symptoms. And then lastly, to provide a little bit of insight into what preclinical research can tell us about how these psychedelic compounds are exerting their beneficial actions in human disease. So 2016 was the annus mirabilis for psychiatric use of psychedelic compounds. These 3 very fundamental studies came out really to inaugurate the modern era of work in psychedelic drugs, which had really been shut down in the 60s by the war on drugs in the US. had brought a lot of promising work to a halt. And really, with these 3 studies, was the first time the government had relaxed the controls in the U.S. so that in England, so that careful clinical studies could be performed looking at potential benefits here. And so these are the 3 major papers dealing with sort of depression-related issues, both treatment-resistant depression and also depressive symptomology associated with end-of-life issues for patients with life-threatening cancers. In the meantime, there's also been any number of other psychiatric indications in which psychedelics have been shown to exert beneficial therapeutic actions. But really, I'm going to keep my eyes on the depression field for the most of this talk here. So there's been developed through this work a sort of a standard protocol that's used in essentially all of the academic clinical trials that have been done to date. And it involves a series of 3 visits. The first visit is maybe an hour or 2. The subject comes in and they meet with the facilitators that will be with them while they undergo their psychedelic experience. They prepare the patient for what they're about to experience. Anybody who's ever had a psychedelic compound will tell you that the profound alterations of consciousness and perception are among the most spiritually meaningful and also very powerful experiences they've ever had in their lives. And so it's been found that it's important to prepare people for what to expect while they're going to take their psychedelic drugs. Then a day or so later, they come in and they have their dosing day, their treatment day. And so this is a typical setting. These are nice, comfortable rooms, not cold hospital rooms. The patients are given a relaxing setting. There are soft music, eye shades, soft lighting, and they consume their psychedelics, almost always the psilocybin in these trials, and they're told to concentrate on the inner experience, that what they're feeling, what they're thinking as they undergo their psychedelic experience. And it's been found that if you prepare people for this psychedelic setting and you provide a very comfortable setting, that people generally have a very positive experience. There's none of the sort of the bad trip experiences that people had in the 60s maybe. And so on the whole, these trials have shown a great deal of safety and a great deal of acceptance for the subjects. And so they experience what they experience. There are 2 facilitators that sit with them during this trial in case any strong emotions come up, any anxiety, any fears. They're there to help calm people down and to make sure that they feel safe during this process. But there isn't a lot of direct therapeutic interaction here. The facilitators are not asking questions about the patient's lives, their parents, their childhood, those kinds of things. And they're just told to experience what they experience. Then they come back in after the drug treatment day. A couple days later, they come in for an integration session. And again, this is only an hour or 2, of discussion of what the experience was. But this is very much a pharmacological treatment and very minimal, as low as one can go, I would say, direct sort of therapeutic intervention with the patients. And that's deliberate design. This is maybe not the best way to make people get better, but it is the way to sort of understand what the drug itself is capable of doing in this kind of a setting that may have beneficial actions. So these are some typical results from these early trials. And so this is the Carhart-Harris from 16. You can see here self-reported depression scores on the y-axis. So a high number is a lot of symptoms of depression. Low numbers are good. You can see the baseline levels in these patients here. They come in, they're given psilocybin, and within days to a week, you can see this very large and very statistically significant improvement in their depression scores. And these scores retain their improved psychological state for months, even in this study out for a full year after they've been dosed with a single administration of a psychedelic compound. So this is really why we're all excited about using psychedelic compounds. These are very large effects by the standards of standard antidepressant drug treatments like Prozac and SSRIs, and also very persistent. They're also very rapid. SSRIs, you probably know, take weeks and weeks to have an effect if they are going to have an effect. And so this has gotten people really, really excited about the potential of using psychedelic drugs to treat psychiatric disease. So great, why aren't we just opening up clinics on every street corner and every hospital and treating patients every day? There's a lot of obstacles that need to be overcome before we can really harness this potential of psychedelic medicine for treating psychiatric disease. Some of these obstacles are sort of scientific, and this is a really important one. Does it really work? These are patients that are suffering from severe depression. Most of them have suffered depression for many, many months and many, many years. They come in, they've read the newspaper, they've seen the movies, they've talked to their friends. They're all convinced that psychedelics are the exciting form of medicine. This is going to finally be the thing that makes them better. So they bring in a lot of expectancy that this trial is going to work. And so there's a very strong reason to suspect that there's a very important placebo effect in these kinds of studies. In particular, because it's impossible to blind the subjects to what kind of medicine they've been given. If you've been given a psychedelic, there is no other drug that you can give the patient that they would confuse with the profound alterations in perception and consciousness that psychedelics produce. And so that means the patient knows exactly what they've been given, and there's no way to prevent that. And lastly, we pretty much rely on asking them how they feel as our endpoint, right? And this is a weakness of psychiatry as a whole. And so if you have this combination of not being blinded and having strong expectation that this is going to work, if you are the subject, there's a strong chance that you are going to report that you feel better. And this was a very concerning study for that, to me. This was a comparison of the effects of psilocybin. So this is, again, change from a depression score. Small change is bad, big change is good. And they did a direct comparison of psilocybin and escitalopram, which is a classic antidepressant drug treatment. And what you can see is that within the first reporting period, so the first week after treatment, the patients with psilocybin reported an improvement in their score. That's good. But the patients that got the SSRI here also reported an improvement in their score. And that is almost surely not the real effect of this citalopram. And in fact, these two data points here are not significantly different, which again sort of highlights for me the risk that many of these effects of psilocybin could be related to sort of a placebo effect of expectancy. There are also some practical obstacles. So of course, psychedelic compounds produce this massive effect on consciousness and perception. That means that you need to have a full day in the doctor's office, plus that pre-day and the post-day. You need to have two people sitting there. That's at a huge economic cost, both for the care providers and also for the patient. They have to take 3 days off from work. They have to have somebody come pick them up and drive them home. So that's an expense, and that cost is going to prevent these compounds from ever, I would say, reaching their full potential to treat the large number of patients that could benefit, because there are few that will be able to afford this expense. This is an issue for getting the FDA and the public to accept these kinds of medications as well. A lot of the public is rightly scared of the intense experience that you have with psychedelic compounds. These compounds that have been used so far have a relatively prolonged time of action, so about 8 hours. So that means, again, you have to sit there for the full day in the doctor's office, and the facilitators have to spend the full day with you. And lastly, there is a risk that because these psychedelic compounds produce a hallucinatory response that is very similar to the psychotic hallucinatory responses, there's been a lot of fear that use of psychedelic compounds in subjects that are at risk for developing psychotic symptoms could provoke a psychotic break in those patients. And so all of the trials that have been done to date have been very careful to screen out patients with any family history of psychotic disease. And so insofar as these medicines become more widely available, there's a greater risk that people will slip through that safety net. So the question then becomes, how do these drugs work? And of course, the dominant idea is that because these compounds produce such an intense alteration of perception and experience, and are reported by everybody who's had a psychedelic compound to be amongst the most spiritually and emotionally meaningful events in their lives, there's a strong assumption that this experience is what leads to the beneficial action. And so this is a study from the group at Johns Hopkins Universities, both in a smoking study. So here, lower values here mean less smoking. And this is a depression score. Again, lower values mean stronger antidepressant effect. And what they plotted here was a correlation between the intensity of the alterations in perception and the benefits that the patients experienced in reducing smoking or reduced depression symptomology. And so these kinds of correlations are observed, but of course it's not a direct proof that they're actually linked. Because, of course, the more receptors that the drugs bind to, the more you're going to experience this, and the more you're going to see perhaps a beneficial effect. But it doesn't establish cause and effect. So is this psychedelic experience really necessary for the beneficial actions of the psychedelic compounds? So this remains unknown because we still don't really know how these drugs work. What do they actually do that underlies this beneficial action? So all psychedelic compounds are potent. All the classic psychedelic compounds, LSD, psilocybin, mescaline, are all potent agonists at serotonin receptors. So here's an example for psilocybin. Psilocybin is actually a prodrug. When it's ingested, it's rapidly dephosphorylated and converted to psilocin. Psilocin is the active ingredient that circulates. And you don't need to be a neurochemist to recognize that this structural compound psilocin is very similar to the structure of serotonin, the natural ligand for these serotonin receptors. There are 19 different serotonin receptors. Is that right? 14, 19, a lot of serotonin receptors. And these classic psychedelics have appreciable affinity for almost all of them. And in fact, in some cases, they have higher affinity than serotonin itself. Some cases, the lower affinity than serotonin itself. And so one of the questions that remains to be answered then is which one of these receptors that's activated by these psychedelic compounds is the one that's important for the therapeutic action. We know that one receptor in particular, the 5-HT2A receptor here, is the one that mediates the alterations in perception and the psychedelic experience itself. And we know that because trials have been performed in human subjects in which psilocybin and LSD are administered together with ketanserin, which is a pharmacological antagonist of the 5-HT2A type serotonin receptor. And when you block the 2A receptor, then LSD and psilocybin no longer produce any psychedelic alterations in perception and consciousness. So again, the 2A receptor is mediating these effects. What we still don't know is whether the 2A receptor is necessary for the beneficial actions of these compounds against depression, smoking, addictions, and other psychiatric diseases in which they have benefits. So that's an open question. There are a lot of approaches that are being developed to try and dissociate the psychedelic response from the beneficial action. Again, the thought here is that if we could reduce the perceptual alterations, the psychedelic response itself, then these compounds could be safer, more easily administered. You wouldn't need two people sitting in the office. In fact, if you didn't have the psychedelic trip, you could probably drive home after your doctor administers the drugs. That would increase utility. That would make it easier for the public to accept using these medications to treat their disease. It would lower the cost tremendously and therefore increase the potential benefits. So several strategies are underway to try and minimize the psychedelic effect and hopefully retain the beneficial actions. Most of these studies are still at the level of preclinical, and we still don't know from clinical studies, but one of the things that is working on, that people are working on, is developing so-called biased agonists that activate the 2A receptor but don't produce the signaling mechanism pathways that are underlying the psychedelic effect. Those are thought to be sort of classic G protein mediated pathways, but people are working on biased agonists that are more potent for the other beta-arrestin signaling arm of the pathway. And again, at least in mouse studies, those non-psychedelic psychedelics seem to exert actions that might suggest they would be effective drugs in people. Again, remains to be shown. Other people are working on developing alternative psychedelic compounds that have a shorter mechanism of action. So DMT is the favored one here. A DMT trip lasts about 2 hours as opposed to the 8 or more hours for other classical psychedelics. So again, the thinking here is if you only needed to sit in the doctor's office for 2 hours, maybe you could just make this whole process faster and easier. And then you're probably aware that another strategy that's incredibly popular with the general public is the idea of microdosing. That is, you take a dose of a classic psychedelic like LSD or psilocybin, but you take it at a dose that is low enough that it does not produce a detectable alteration of your perception and consciousness. And that people report that this microdosing has beneficial actions. So that sounds good, but when careful studies have been done in academic environments, you get a very different kind of result. So this is a study that was published in 2021. These are various measures of response to psychedelic compounds. In this case, a high number is good and a low number is bad. In this case, the high number is bad and the low number is good. And so here you can see in black the drug that they were given, either placebo or a microdose. And what you can see here is that people who got the placebo and the microdose both reported a beneficial response to this drug, and same over here. And in fact, what really determined the way the patients responded was not what they actually got. Some people that got the microdose didn't get better, some people did. But what matters is what they thought they got. And so again, these are microdoses. They don't produce a detectable response. But if you think you got the microdose, then your score is improved in both of these measures. If you thought you got the placebo, then your scores did not improve. Right? So it didn't matter whether you got the placebo or the microdose, it only matters what you thought you got. So this evidence, and there are several other similar studies, sort of suggests that microdosing is a very complicated, expensive way to fool yourself, right? Which isn't to say that it isn't effective, because the mind is a powerful thing, but it does not suggest that it's a pharmacological response to the drug. Nevertheless, there are people doing it every day. So what can we learn from preclinical research that can provide some sort of information and inform the further clinical development of psychedelic drugs? And so this really speaks to the mechanism of action, and in order to know how these things work and produce an antidepressant-like response, we ought to have some sort of idea of what it is we're trying to fix, that is, what is wrong in the depressed brain, and how do compounds that we know are effective antidepressants work, and do they then share similar mechanisms of action with psychedelics? This seems to have gone dead. So I'd like to just take a little bit of an aside here and talk about how, from a big picture, how we think about what is wrong in depression and how antidepressants work. And so depression, as you know, is characterized by a wide range of symptoms, which suggests that many regions of the brain are affected. If you have a loved one or yourself are brought to a doctor, there are 2 symptoms that you absolutely have to have in order to receive a diagnosis of depression. One is obviously prolonged depression in your mood. That's what we all think about when we think about depression. And the other is anhedonia, which is the inability to derive pleasure from things that should be pleasurable, like celebrating the anniversary of the UNAM and listening to speakers come from all over to entertain you. If you are depressed, you will not find these lectures entertaining. So I'm not saying that anything here, and then there are a whole host of other symptoms you may have. You may lose sleep, you may lose motivation, you may lose appetite, cognitive problems. Again, suggesting that large regions of the brain are affected. And we know also a little bit about the etiology of depression, and that is that there's an interaction between genetics and the environment and your developmental stage. We know from genetic studies, people keep trying, but what they find repeatedly is that less than half of the risk for depression can be defined by your genetic makeup, at least so far. It seems like there are many genes that can contribute small amounts of risk, but no particular depression gene. Environmentally, on the other hand, we know that one of the greatest predictors of who is at risk for experiencing depression are stressful life events. These can be sort of the things that come first to mind: physical illness, problems with your family relationships, losing your job, losing your health insurance, losing a loved one. All of these can be, sort of associated with risk and in actual incidence of an episode of depression. And it's important to remember that there are also other things that are psychosocial stressors. They are stressful, but they aren't so easily correlated with something in your environment. But people who are at risk for depression tend to think more negatively about things happening in their lives. They hold the negativity in them. They have a bias in their perception of things that are negative compared to positive. And that is, again, a risk factor. We also know that there are other things that are stressful, and one of the biggest correlates we know is chronic pain. People who are suffering from unrelieved chronic pain are very at risk for depression. We also know that there's a lot of these environmental issues that when they happen early in life are very strong predictors of a wide range of health consequences, and depression is included amongst those. So what, again, what does this do to the brain? How do we think about the brain? And we know a couple of things. We know that there are anatomical changes that can be observed. And so there is an atrophy that is associated with the hippocampus in particular, was the original description. And people with strong changes, atrophy in their hippocampus, are less likely to respond to antidepressant drugs. The same is true for the prefrontal cortex, an area that's important for a lot of executive cognitive functions. Various prefrontal cortical regions shrink in human depression in imaging studies. And even chronic stress in healthy individuals. This is a fascinating subject from Liston, study from Liston et al. They took healthy subjects into the clinic every day for I think it was a month, and subjected them to a variety of chronic stressful behaviors, solving math problems in front of other people, standing up and speaking to an audience. And what they observed was that even in healthy subjects, if they were repeatedly exposed to these stressful tests, that they also experienced an atrophy and a shrinking of their prefrontal cortex here. So stress is bad for your brain anatomy. And not only are the areas themselves shrunk, but more recent studies now looking at functional connectivity using fMRI have established that these same brain regions that are associated, that are atrophying, are also then less tightly connected to the other regions here. And so the idea is that when these regions are shrinking, there is damage to cells. Oops. And this is what changes the way these brain regions can do their job, right? They are not, the cells are not healthy. That means they're not communicating properly with each other, and that is impairing the function of these brain regions. And these are changes that we can mimic in our preclinical mouse models here. So in my lab, we use a variety of forms of chronic stress. It doesn't really matter what you do to a mouse or a rat, but several weeks of something that is chronically stressful, either fairly physical or fairly psychosocial or even purely neuroendocrine, will produce changes in behavior that resemble these symptoms of human depression, right? So what are the behaviors that we should use, right? Is that a depressed mouse there, or is that a wistful, thoughtful mouse, deeply engaged in thinking about his relationship with his parents? We need good behaviors, and there's a variety that people use. I'm going to start with the not so good ones, in my opinion. These are typical escape behaviors. So for swim tests, tail suspension tests, they are what they sound like. You literally tape the mouse to a cabinet in the laboratory by its tail, and you measure how long it struggles. And they are thought to be measures of sort of helplessness, right? Despair. Right? If I'm depressed, I'm going to give up easily in the face of adversity, and that's what it's thought to be monitored here. But of course, there's no way to know what the mouse is actually thinking when it stops struggling at the tape to your cabinet. And there's some other ones here. So these are fine in the sense that they're sensitive to chronic stress. You can measure a change in this behavior, even if you don't know what it means. And there's evidence that they do respond to compounds that we know are pharmacologically active in human depression, such as ketamine, for example. But the thing that, in my mind, makes them not very good tests is that if you give an animal an SSRI like Prozac, they immediately respond in these kinds of behaviors. Within minutes, there is a change in the behavior when you inject with fluoxetine, for example, Prozac. And that is something that we know does not happen in human depression. People do not respond immediately to SSRIs. It takes many, many weeks before their behavior improves. So that, for us, is the first piece of evidence that maybe these are not good behaviors to look at. And so in my lab, we like to look at things that provide a readout of anhedonia, because you just can't ask the mouse whether it's feeling sad. At least you can ask it, but you don't get an answer. And so hedonic state, one of these cardinal symptoms, again, is something that you can ask the mouse about. You can give it various forms of rewarding stimuli, and you can ask how the animal behaves in response to a reward. And I'll show you some examples of what this looks like in a bit. These are also sensitive to chronic stress. If you take an animal and subject it to chronic stress, it develops an anhedonic state. It no longer responds normally to rewarding stimuli. These behaviors are also respond immediately to fast-acting antidepressants like ketamine. And importantly, and really the thing that made us choose these in the beginning, is that when you give them an SSRI, they will respond, but only if you administer the SSRI for several weeks. You don't get that immediate response in these behaviors here. And I'll show you an example of that too. And lastly, there's some face validity here, because these are behaviors that are very directly tied to the human symptom of anhedonia. So what do we know about chronic stress in preclinical models? Well, the work really began in the lab of Bruce McEwen at the Rockefeller Institute in the US. Who was the first to describe this atrophy of hippocampal and pyramidal cell dendrites and a loss of dendritic spines. And so they speculated then that this atrophy of the cells can account for that shrinking of overall volume of the hippocampus that you see in MRI studies. And it also predicts that there will be functional changes in the way these cells interact with their neighbors via synaptic connections. And we went on to actually test that possibility in hippocampal brain slices. And so what you see here on the left are recordings of synaptic strength measured extracellularly in brain slices. So we take animals, we subject them to chronic stress, we bring them down to the lab, we put their hippocampus in a dish, and we can measure changes in synaptic strength. And what we observe is that chronic stress, but not acute stress, produces a decrease in the amplitude of these excitatory synaptic currents that we measure in their pyramidal cells here. In particular, those changes are localized out in the distal dendrites, which is exactly where McEwen's group reported the anatomical changes. We know at least one contributing cause for this weakening of synaptic function is that there is a loss of synaptic proteins, in particular AMPA receptors here, the GLUA1 subunit goes away in parallel with the loss of synaptic function. And if you give animals an SSRI like fluoxetine, chronically but not acutely, you can get a restoration of both synaptic function and AMPA receptor expression. So chronic stress, bad for your synapses, bad for your brain cells, your neurons, and these kinds of changes can contribute to the functional impairments that these brain regions are going to be associated with. Very similar findings in the prefrontal cortex. This is work from Jason Radley. Again, looking at the dendritic morphology of pyramidal cells, there is an atrophy, a loss of dendritic branches, loss of dendritic spines, and then a correlating functional change in the strength of the excitatory synaptic projections onto these PFC pyramidal cells here. We've also looked at the nucleus accumbens, which is an important regulator of dopamine release back to the forebrain. Dopamine, of course, is the neuromodulator that makes us work for reward and has a lot to do with how rewarding we find various stimulus to be. And what we observed is that with chronic stress, there is a loss of excitatory, a decrease in excitatory synaptic strength onto the MSNs of that express the D1 dopamine receptor, which are the positive rewarding cells within the nucleus accumbens. And just like in the hippocampus, if you give fluoxetine chronically but not acutely, you can restore the function of these synapses onto the D1 cells here. So taking all these, and again, if we look at reward behaviors that are associated with and what chronic stress does to them, what you can see here on the left is the sucrose preference test. So this is a 2-bottle choice. The mice have to choose to drink from plain water or water that contains sucrose. At baseline condition, 80–90% of all the fluid they drink will be from the bottle that has the sucrose in it because it tastes good, it's rewarding. If you subject them to chronic stress, what you find is they drink equally from both bottles, so they've lost that desire to preferentially drink from the rewarding bottle. If you give them fluoxetine, again, chronically but not acutely, you can restore that preference that they have for the sucrose-containing solution. Similar effects in cognitive tests, cognitive flexibility in particular, and also conditioned place preference, which is, again, a measure of the animal's willingness their ability to respond to a rewarding stimulus. They like the reward at rest, they lose the response to the reward after chronic stress, and then chronic antidepressant treatment will restore that reward behavior. Similar things are starting to become apparent in human imaging now too, so this is one of the best studies to date. This is looking at functional connectivity between the prefrontal cortex and the ventral striatum, the nucleus accumbens. And what you can see here is the improvement in patient scores on the y-axis when they've been treated with escitalopram. And so nope. A high number here means a strong improvement. That means the antidepressant treatment worked very well. And a low number means a poor response to the antidepressant. And what you can see on the x-axis then is the corresponding functional connectivity between the prefrontal cortex and the nucleus accumbens. So the stronger those connections are functionally, the bigger improvement that the patients experienced in their response to the antidepressant drug, which strongly suggests that the two are related functionally. So putting this all together, again, we have the idea that there are these structures that are important for detecting rewarding stimuli in the environment, like the hippocampus and the prefrontal cortex. They speak to each other. They also speak with excitatory synapses to the nucleus accumbens. The nucleus accumbens is a regulator of dopamine secretion from the ventral tegmental area and gives the signal that this, to the animal that this is a rewarding, thank you very much, a rewarding stimulus that they've experienced, and dopamine comes back and says, do that again, do more of that, keep at it, that's a good thing to do, right? And so the idea that we have then, the hypothesis is that when you experience chronic stress, you have a weakening of excitatory connections within these brain regions and also between these brain regions. That results in a decrease in the release of dopamine back in response to the rewarding stimulus. And that is what accounts for the anhedonic state that characterizes human depression as well as the response of mice to chronic stress. So if that's what you think is wrong, then what you want an antidepressant drug to do is to first restore those excitatory connections back within these brain regions. And by restoring those excitatory connections, you can restore then the ability of the nucleus accumbens to trigger dopamine release back up into the forebrain here. So this hypothesis predicts then that anything that has an effect on excitatory synaptic strength, strengthens excitatory synapses, will have an antidepressant action. Conversely, anything that has an antidepressant action, which you might know first, probably has an effect on excitatory synaptic strength. And so we know that this is true for SSRIs, as I've shown you. It's also true for various compounds in development, ketamine metabolites, GABA NAMs that Teresa mentioned before. And it really suggests that this is a common feature of all compounds that are antidepressant. So with that as a background now, how can we think about the ability of psychedelic compounds to exert their beneficial actions here? So when we started this work, first of all, an unanswered question was, do these compounds actually have beneficial actions in mice, right? If you think that psychedelics work because they're causing this great internal conversation and your ability to rethink your relationships with the obstacles in your life, your parents, your children, your friends, then maybe you might expect that these compounds wouldn't really have an action in mice, right? So is there anything in mice that these compounds do that looks like it has a relationship with the antidepressant response? If you have that, then you can ask what changes do these psychedelics produce in the brain? And in particular, we also asked, is this 2A receptor necessary for these beneficial actions in mice? 2A receptor, again, is the one that mediates the alterations in perception and consciousness. So in thinking about the problem, it's useful to sort of break it up into two phases. So this is the graph I showed you before. This is a Beck's depression score here. High numbers bad, good numbers, low numbers good. And you can see that when the drug is on board, it produces this rapid antidepressant improvement in scores, and these benefits persist for many weeks and months afterwards, right? And so in trying to understand how this comes to be, it's useful to break up the problem in two phases. First of all, there's something that happens while the subject has the psilocybin present in their body, right? It's doing something. It's activating serotonin receptors. They're turning on signaling pathways. Some sort of process is induced while the drug is present. And whatever gets turned on here in that point leads to some lasting improvement in brain function that underlies this maintained expression of benefits long after the drug is cleared from the body, right? So again, the things that we've looked at so far is what happens during this induction phase, in particular what serotonin receptors are necessary, and again, what sort of changes can that serotonin receptor activation produce that underlie this lasting improvement in mental health. And so we had the hypothesis that we would in fact be able to detect a beneficial action if SSRIs work by producing serotonin receptor activation, then perhaps the psychedelics should too. We again thought that restoration of excitatory synaptic strength is a critical potential mechanism that could underlie this antidepressant response. And in particular, based on work we had done with SSRIs, we did not think that the 2A receptor would be necessary. Our SSRI work favors the activation of other serotonin receptors as critical to the antidepressant response. So these are what we set out to ask in mice. So we start out by measuring behaviors at baseline. We subject them to chronic stress. We measure the effects of the stress on the reward behaviors. Then we pre-treat them either with an antagonist of the 2A receptor or with saline, administer the psychedelic compound, in this case psilocybin, and then measure behavioral response to that treatment with psychedelics here, and then bring the animals up, prepare the brain slices, and measure the strength of their excitatory synaptic connections. So first, do psychedelics have a beneficial action in mice? They do. And so this is data here on the left looking at this sucrose preference test again. At baseline, they have a strong preference for the sucrose-containing solution. After chronic stress, they lose their preference for the sucrose solution, and they drink equally from both bottles. And if you just treat them with a vehicle, there's no significant change in their response to the rewarding solution. But if you give them psilocybin, now you can get within 24 hours a restoration of the normal high level of sucrose preference here. So that's good. That is an antidepressant, or at least an anti-anhedonic response to psilocybin in mice. That's good. Same thing is seen here in this test. This is a female urine preference test. So these are all male mice. You give them a choice between a Q-tip dipped in urine from females in estrus and a water-dipped Q-tip. And again, at baseline in the stress-naive situation, they have a strong preference for interacting with the Q-tip dabbed in female urine. They lose that preference after chronic stress, and administration of psilocybin can restore that preference for the female urine. Again, an anti-anhedonic-like behavioral response. So now, does this response require activation of the 2A receptors? And so now you can see here the experiments in which the mice were pretreated with the 2A antagonist ketanserin. And what you can see is that in green, there is both a significant restoration of sucrose preference and also female urine preference here at 24 hours after a single administration of psilocybin in these mice. So that really suggests then that it is not a 2A receptor dependent anti-anhedonic response in mice. So what about mechanism here? Again, the hypothesis was that it had to do with restoration of excitatory synaptic strength. And so if you compare the strength of synapses in animals subjected to stress and not given drug, what you can see here in black is the weak synaptic response. And here, by comparison, brain slices from animals given psilocybin, you can see that the responses are larger. That's a sign of good, strong synapses. You can quantify that by measuring the AMPA to NMDA ratio. And psilocybin produces a statistically significant increase in synaptic strength. So that's good. That was consistent with our prediction and can explain perhaps the beneficial behavioral response here. And just like in the behavioral response, what we find is that animals given ketanserin before psilocybin to block the 2A receptor there show every much as bit of a good response to the psilocybin, a strengthening of synaptic strength here as the animals given psilocybin alone here. So just like the behavioral response to psilocybin, the synaptic response to psilocybin does not seem to depend on this 2A receptor that mediates the psychedelic effects. So we still don't know which receptor is required. Our favorite hypothesis is the 1B receptor, but we're on the path to tackling that question. But the fact that the 2A receptor is not required, again, leads to the possibility, that you could develop psychedelic medicine-based approaches in which the patient would not have to undergo this intense psychedelic response. There's corresponding to our physiological evidence of improvement in synaptic strength. There's also morphological evidence. And so this is data from Alex Kwan's lab looking at dendritic segments here, and each one of these little things you see protruding from the dendrite is a dendritic spine. That's the postsynaptic side of an excitatory synapse. And what they found was that in stress-naive animals even, psilocybin promotes a persistent outgrowth in the number of these dendritic spines. Again, because each of these is an excitatory synapse, that is also an increase in excitatory synaptic strength, and that's what they measured physiologically. And in Alex's work, what they found was that this increase in dendritic spine outgrowth also was not blocked when the 2A receptor was blocked. So this is the outgrowth here, very comparable to the control outgrowth. So that suggests, consistent with our results, that this anatomical change in synaptic function in structure is also independent of the 2A receptor here. That said, there are some other groups that have done comparable experiments and have obtained a different result. And so these are looking with selective 2A agonists. They see outgrowth, and that outgrowth is absent in mice that are lacking 2A receptors. And this is using some of these biased 2A receptor agonists that I talked about before. They also see spine outgrowth here. So this question is still open. Ultimately, the experiment has to be done in human subjects to really know whether it's true or not. And those experiments are underway in several groups right now. So stay tuned for the answer to that. But again, the hope is that that if you can induce a change in people's behavioral scores in a lasting way that is independent of 2A receptors, that you can increase the utility, the acceptance, the economic benefits of this kind of a therapeutic approach for psychiatric diseases. So again, there's still a lot we don't know. We haven't yet figured out which receptor is the critical one, what sort of intracellular signaling pathways are turned on. There's been a lot of interest in BDNF tract B signaling. Eero Castron's group in Finland has suggested that in fact psychedelics directly promote activation of the tract B receptor. Again, this is a really prominent neurotrophic signaling factor in places like the hippocampus and the cortex, and that alone could promote restoration of synaptic function there. There's evidence that various transcription factors turn on that could produce gene-dependent effects that also underlie this lasting improvement in depression scores. Again, I've shown you the evidence that psychedelic compounds lead to rapid and lasting changes in synaptic strength, in the outgrowth of dendritic spines. There's also evidence that they actually promote the formation of new dendritic branches as a whole. In cortical studies, I guess I'll show you in the next slide, there's increase in functional connectivity between brain regions. All of these things directly counter known symptomatology of human depression. So that really suggests that these are, in fact, the causative changes that underlie these persistent expression of beneficial actions here. So this is a study done by the Hopkins group, again, looking at functional connectivity in different brain regions here by color. And what they report is that even a week and a month after treatment with a single administration of psilocybin, that you can detect lasting functional changes in connectivity between brain regions that are important for symptom domains of depression, such as cognition and reward. And so again, these ideas really are offering a potential novel path forward for increasing the utility. And so there are companies such as Terran Biosciences that are very heavily involved in trying to develop non-psychedelic approaches to psychedelic therapy. So just to summarize then, our findings suggest that what happens in depression, and certainly in mice and probably in humans as well, is that chronic stress and other precipitating factors in those individuals that are susceptible to these kinds of changes affects the function of excitatory synapses, particularly within these cortico-mesolimbic reward pathways. When the synapses are screwed up, then the function of the circuits are also screwed up. The brain regions aren't talking to each other properly, and that can give rise to all of the sort of various symptom domains that characterize the human depression. In particular, our evidence from mice suggests that anhedonia and perhaps negative cognitive bias and depressed mood can also be explained by these changes in the functional behavior of these circuits here. Psilocybin, like other known antidepressants, has the ability to restore both reward behaviors in mice and also changes synaptic strength in strong parallel with those changes in the behavior of those synapses in which the circuits in which those synapses are embedded. And that can provide a substrate for lasting beneficial psychological actions of these psychedelic compounds. Lastly, the 2A receptor mediated classical trip of the psychedelic compounds may not be necessary for the antidepressant action that they exert, and this really offers the possibility of increasing the utility of these approaches for treating psychological disease. So I just want to thank the members of my lab that were involved in this work. All the work was done at the University of Maryland, and we are now in the beautiful city of Denver, Colorado, and this is literally the view from our laboratories out looking at the Rocky Mountains, and we will be carrying forward this work, and in particular now, very happy to be in the Department of Psychiatry, and we are about to start enrolling patients for our first human trials using psychedelic compounds. So thank you for your attention. I'm happy to take questions, and I am hard of hearing, so if you get to a microphone, speak loudly and slowly, please.
Hi, thank you very much. You show in your mice that some of them, they were not so susceptible to depression. And I was wondering whether there are studies about maybe the expression of the serotonin receptors, and if there is a correlation in humans, because some of us are not also so susceptible.
Yeah, this is, so we know this from human epidemiology, and this certainly seems to be true in the mice as well. Not all of them respond to the chronic stress in the same way, and then even those that are susceptible, most of them, but not all of them, respond to various antidepressant treatments. And so it's really a challenge to know where that difference in susceptibility arises. I mean, these should be genetically homogeneous populations of mice, so genes seem unlikely to be important, but epigenetic changes. We're going to hear from Tracy Bale on Friday, and she is developing an approach in which we use chemogenetics to directly activate the stress pathway in the paraventricular nucleus. Are you going to talk about that? No, okay. And the idea is that perhaps the hope is that we will achieve a more controllable and consistent stress in that way, and that a lot of the resilient susceptible has to do with more perception of the environment and of the stressor at sort of cortical levels, and therefore less an effect at downstream levels. And by directly activating the PVN, we should be able to reduce the amount of variability there, reduce the number of resilient animals. But we don't know yet. Great question.
I wanted to ask if you have any indicator to know if the dendritic growth might be aberrant?
Sorry, I'm sorry, do we have evidence of what?
I wanted to know if you have any indicator that the dendritic growth that you have with psilocybina might be aberrant? Or is it, or how do you know that it's not dangerous, that the growth might affect other structures? And
yeah, so I mean, this is a very important question right now. So, to what extent does neuroplasticity contribute here? I showed you what evidence I have. There's a lot of people that are very interested in looking at using psychedelic compounds to treat conditions where we know there's functional pathology, so stroke, traumatic brain injury, things like that. There are people and companies very dedicated to testing psychedelics. I think we don't yet have the evidence yet. And what we also don't have is a direct link between the morphological plasticity and the functional plasticity and the therapeutic action, right? In principle, you should be able to go in and block some signaling pathway and prevent morphological plasticity. That should prevent functional plasticity. That should prevent behavioral plasticity. And we don't have those manipulations yet. Yeah, important question, though.
thank you.
You're welcome.
Yes, thank you for a very interesting talk. I want to ask you whether there's any relation with the beneficial effect of psychedelics in terms of sex or age.
So the clinical studies to date have not found any important sex differences. So males and females respond equally well to the psychedelic therapy. And then we're up now with all the careful clinical studies have been done. we're probably up in the order of 500 patients that have been treated so far. I think the response rate is something like 80%, and then that's equal for men and women.
Thinking that hormones, steroid hormones may make a difference, or reproductive state as well.
It's certainly going to be a factor, but right now there's no evidence that I'm aware of that speaks to that question directly. And you asked about age as well. I think, all the studies to date have been done on, I'm going to say, 18 years or older, right? I mean, we're all very leery to test these powerful mind-altering substances in preteens or teenagers, I think. Again, the risk of triggering a bipolar episode is holding us back.
Thanks.
Yeah.
Hi. I really liked your talk, and
Thank you.
I have 2 questions. The first one is regarding the methodology of the therapy, where you close their eyes and you put music. What type of music do you put?
You can go to Spotify and you can download the Johns Hopkins psychedelic trial playlist, actually.
Okay. And
I have to say I haven't done it, so I don't really know. I think the idea is you want the patient to be relaxed, but you also don't want to distract them, right? And you don't want you want the music to be sort of in the background as a way of reducing external distraction. I think, let's put it that way. So most of the music is instrumental, so no spoken word for the most part, or sung word. It's very low key and
okay.
Yeah.
And because there are a lot of different genres that that are only instrumental, how did they choose which music to play?
Yeah. You should go on Spotify and see if you can find that. I've been told it's there. I haven't actually tried it, but yeah, that is the thinking, is to help people be in a relaxed place where they can concentrate on their internal state.
Okay, thank you. And the second question is kind of maybe not really related, but it came to me when you spoke about chronic pain and how that can induce depression. And specifically about fibromyalgia, which is like a disease in which the pain and the depression are really closely related. So if you have more pain, you're more depressed. If you're more depressed, you have more pain. So do you think that fibromyalgia could be treated with psychedelics?
Yes, I believe there is a good clinical trial that's been formed that does show benefits in fibromyalgia. A lot of benefits also in cluster headaches, which is, I've been told, one of the most debilitating things that you can have. So yes, there's, pick a disease, and there's starting to be evidence that psychedelics work, which is a little bit suspicious, perhaps, but that's where the hype and hope comes in, I guess. But I'm pretty sure there's a good fibromyalgia study, and other forms of chronic pain as well.
Mm-hmm. Yeah, thank you.
Yeah. And I think the idea there is, again, the sort of idea that you're promoting neuroplasticity, you're reforming connections, you're breaking down hardwired ways in which you continue to perceive anything that's happening in your environment as this is going to hurt, right? And if you think everything is going to hurt, then it does. Right. Hi. Oh, yeah.
Thank you, Doctor, for such a great talk.
Thank you.
Actually, it beckons a lot of questions from me, but I'm just going to be...
I'm around all week, so come find me.
First off, this is like a very interesting talk because it compounds the effects, it compares both pharmacological approaches like escitalopram, which is often used for depression, and alternative medicine, which is psilocybin. I was quite impressed that you did this precise comparison, because in a battery of patients, in a prospective study of patients with depression, some of them use psilocybin to see their effects. And it's something that, the main question that I want to ask you is, alongside whatever effects or whichever actual receptor is activated with the psilocybin, is there a psychotherapy associated to increase the effectiveness of both escitalopram or psilocybin that you've explored?
No. So I think where we are in the development of this modality of treatment, we know for psychiatric disease that talk therapy works, right? If you have 2 hours a week and $500 an hour to pay your therapist, you will get better eventually. It might take you 10 years, it might take you a lifetime. So we know talk therapy works. Most people assume that anything that psychedelics do to increase neuroplasticity, defined loosely and fuzzily, will increase the ability of you to change your mind, right? In the same way that talking over and over with a therapist allows you to see your problems in a new light. And I think that most people assume and hope that psychedelics will just make that whole process go faster and easier and better and more lasting than the slow, painful slog through normal psychotherapy. And so ultimately, I think most people think that the best way to treat patients is going to be a combination of a psychedelic drug and some pretty intense therapy. Where we are in the development pathway, though, is we want to know, does the pill work? Which pill of all the various pills we might take is the right one to take to push forward? Once we have those kinds of things answered, then we can go through, and what you'd really like to see is a clinical trial where somebody gets psilocybin and very minimal therapy versus somebody who gets psilocybin and maximal therapy, where you're really using the post-integration day to really delve into the person's issues with their family, with their life, with their struggles. And everybody's expectation is that the combination of therapy and psychedelics will be best.
And a second question, will be the last one. You mentioned that there is a glutamatergic input from the prefrontal cortex to the nucleus accumbens, correct? And you also mentioned that the glutamate subunit A1 is involved in it. Do you think that any sort of therapy or stimulus to prefrontal context or an enhancement of the glutamatergic pathway could help enhance the effects of psilocybin?
Right, so the classic way to elevate your dopamine levels is to take cocaine, right? Cocaine inhibits dopamine reuptake, right? And so, yeah, you're gonna feel good for a little while, but it's not an antidepressant treatment, right? So there's more involved than just elevating dopamine. And in particular, what you have to do is to reform the endogenous pathways that allow dopamine to be released, not in a bolus like after cocaine, but after things that are rewarding, things that you should do in your life. So is that a sufficient answer? I mean, raising dopamine is a part of the answer, but not the whole answer.
Thank you.
Hi. So my question is related with humans. I've heard that in a normal treatment, depressed patient, when he starts to take drugs, approved drugs, there is increased risk of suicide. Is there any info about it?
So people in the psychedelic trials have been very, very careful at monitoring suicidal ideation, suicidal thinking. And the evidence to date is that is not such an issue for psychedelic therapy as it is for treatment with SSRIs and other conventional therapies. There's no evidence for increased suicidal thinking in psychedelic therapy.
Oh. Okay, well, that's all.
Yeah, very good.
First of all, thank you for your talk.
Yeah, my pleasure.
Well, I have a really specific question. I have the luck of have read your article, and one of the last articles you have presented, Show It All. And, well, your results are pretty much consistent with them, with the synaptic remodulation stuff, but I always had a question about your article, the Hessler wave.
Uh-oh.
It is, I include both in my bachelor thesis. And I have the question, why choose ketanserin instead of a knockout model or knockdown model? because we know, and so it's something that Xiao et al. Discussed in the paper, that ketanserin only blocks 30% of the receptors. So if you want to account for, block all the effect that receptor has, well, it's not, at least in my mind, it's not the best way to go. So why choosing ketanserin over knockout or knockdown design?
In some ways, it may have been an unfortunate choice, but our thinking at the time was we wanted to recreate exactly what was done in the human trial with ketanserin and psilocybin and the psychedelic experience. Actually, I think they might even be here. Yeah. So we did some controls, because obviously if the ketanserin isn't blocking some or all or enough of the 2A receptors, then it's a spurious result. So I apologize for going off the deep end here, the details. So one of the things that people measure with psychedelic drugs is something called the head twitch response. This is in mice, rotational movement of the head that is correlated with psychedelic potency in humans. In mice, the head twitch response parallels the human psychedelic response. So it's used as a proxy really for 2A receptor activation. And so here are the head twitch data that correspond to the mice that I showed you before. Psilocybin increases head twitch responses, and that is blocked and not significantly different than baseline after ketanserin. So that first suggests that the ketanserin is working. But if you notice, there's a couple of mice that, even though they got ketanserin, still had a high number of head twitches. So you might say, maybe those ones had a lot of 2A activation, even though you thought you injected them with ketanserin. So what we did is we took all of the mice here that had a lot of head twitches, regardless of what happened, what drug they were given, and compared them to all the ones down here that didn't have a lot of head twitch. And the idea is that if you have a lot of head twitch, you probably had a lot of 2A receptor activation, right? And so you might think that those are the only ones that got better, and in fact, these ones didn't. So in fact, what we found was that there was a significant improvement in the sucrose preference and also the female urine preference, regardless of whether there was a few head twitches or a lot of head twitches, right? So there was no correlation. If the head twitch is 2A receptor activation, there was no correlation with the presumed amount of 2A receptor activation and the behavioral response. We also, same for synaptic physiology as well. And we also used EEG recording to verify the effectiveness of the ketanserin. So psilocybin in mice, as in humans, has this ability to decrease oscillations in this sort of delta low frequency range here. That's the blue color after administration of psilocybin here. And when you administer the psilocybin after ketanserin, you essentially eliminate that EEG response to psilocybin here. So that was our other means of saying that this protocol that we used is effective. Is it the best? Would a 2A receptor knockout mouse be better? Yes, we should do that. We should have used we should use a more specific antagonist like volanesencerin, MDL, one of these better compounds.
Thank you so much.
Yeah. Sorry, long-winded answer. Hi.
Hi, thank you. My question is if you know if there's any legal approved psychedelics-based therapy ongoing in this country?
In Mexico?
Yeah, in Mexico.
I don't know what the status is. In the US, we now have 2 states that have decriminalized possession of psychedelic compounds. So Colorado and Oregon State have both decriminalized possession. There's a healthy underground market. You're allowed to possess it, grow the mushrooms, but you're not allowed to sell the mushrooms. I think that's the law. And we are in Colorado, we are licensing, we're working out the procedures to license clinics where you can go and essentially have this same kind of a protocol. You sit with a facilitator and experience your psilocybin in a clinical setting. But Mexico, I'm sorry, I don't know. Of all places, you would think this would be one. Right?
Yeah, maybe. But even in Colorado, there's not like an approved therapy yet?
Not yet, but soon.
All right.
Soon, yeah. Hang in there and then come visit us.
I'm just curious.
I
Thank you.
I think it's supposed to go into effect in January.
Hi, thanks for a great talk. I was just curious about the effects of psilocybin in the mice because how do you measure if they have psychosis, for example, a psychotic event, or those types of effects that you see in humans? And I mean, you basically you filter them so that you don't get this. But what about the mice? Do you filter them too? Do they have trips?
It's a good question. It's something I've been toying with in the back of my mind and haven't really sat down to really put it down. But I believe there are mouse models that have an enhanced risk for undergoing psychotic-like behavioral responses. And so to me, it would be a great experiment to give one of those mouse lines a hit of psilocybin and see, is there any lasting change in their behavior that might make you think about psychotic-like responses. So looking at forms of executive function, short-term memory, things like that.
But what about these mice? Do you have any secondary effects from psilocybin, for example?
No. So we didn't see anything like that, but we didn't do any particular tasks to look for that. Beta Mogadam in Oregon has looked in rats at cognitive processing while the psilocybin is on board, and they, not surprisingly, don't behave very well. But she did not report any lasting effects on cognition. There might be a couple of those studies that I'm not able to recall fully. Mm-hmm. But it's an important question, right? I think it's something we really need to know. If we could develop a biased 2A ligand that would be non-psychedelic, would it still have the same risk for triggering a psychotic break that a true psychedelic compound would have? Mm-hmm. We don't know.
Thank you.
Yeah. Important question.
Hi, we have a couple of questions here. So the first one is, I realized that there are specific efforts to tagging or detecting specific pathways or receptors responsible for these specific effects. So the question is, are there any efforts exploring synergic mechanisms?
Cholinergic mechanisms?
Are there any efforts exploring synergic mechanisms?
Serotonergic. Synergistic.
Yeah. I mean, approaching it not as maybe a single way, but as a complex.
So these drugs are these psychedelics, all the classic psychedelics are very promiscuous. They activate, in addition to almost all of the serotonin receptors, if not all the serotonin receptors, they also have some affinity for transporters, some affinity for adrenergic receptors, some affinity for dopamine receptors. And sadly, the answer is no. I don't think any of those other receptor pathways have received any attention at this point. Oh. It's a really important question.
Yes, yes, of course. And the other question is regarding this presumption that it would be very important for us to get rid of the psychedelic episode or effects of alteration of consciousness and such. So I'm wondering that there are different protocols or just therapeutic approaches to different situations or syndromes that are actually kind of protocols, right? So that you get admitted somewhere or that you go to a bunch of sessions, and actually the current treatment for depression entails such a practice, right? So I'm wondering, why is it worth, in your opinion, to get rid of these kind of spiritual experience?
Yeah, so I mean, I don't want to downplay in any way how meaningful this, let's call it the spiritual side of the psychedelic experience is, right? Anybody who's had a psychedelic will tell you it is the most meaningful, powerful, emotional experience they've had, short of marriage and childbirth, perhaps. Nevertheless, it's an impediment to widespread use. Not everybody can spend 3 days coming to the doctor's office. Not everybody can spend the entire day sitting there. Not everybody will be able to afford to pay for a full day with 2 employees sitting there with you while you have your psychedelic experience. And there's just no way to get around that cost, real economic cost, and make it widely available to the people that can benefit from it. At least that's the thought. And the other thing, of course, is that, well, then why shouldn't I just go out and get some underground mushrooms and have an experience in the privacy of my own home? And so we started jokingly saying this in Colorado, and it's really true. It's ask your doctor if psilocybin is right for you. There is this very strong fear that powerful psychedelic compounds will trigger a psychotic break. If the clinics that are opening in Colorado, if you and your friends sitting around in your apartment are about to experience psychedelics, you don't know whether you're at risk or not. And so you really need to have somebody who will take a look at your family history, will talk through that crazy uncle who was always out in the yard walking around in circles talking to himself. Maybe if you have that kind of crazy uncle, maybe psilocybin is not right for you.
Yes, of course. Thank you.
The last question, please.
Yeah.
Great talk.
Thank you.
I want to ask if the hypothesis is that it restores the reward cycle. Could it be used for the treatment of addiction? And if there are reports of interactions with other drugs?
Yeah, so there's the best studies to date have been for nicotine and tobacco smoking, right? Very powerful, lasting improvements in helping people quit smoking, which is one of the most day-to-day, one of the most impactful addictive compounds we know. And there is a lot of ongoing work going on with other addictive compounds as well, opioid addictions. For reasons that are mostly historical and not so much scientific, to my knowledge, for example, there's been a lot of evidence because of community evidence that exists that ayahuasca may be particularly useful for treating addictions as well. If ayahuasca works, then there's no reason to think that psilocybin and some of these other compounds wouldn't work also, I guess.
Thank you.
Yeah. Thank you very much. Thank you.